Welcome my dear students to Biology at the rate Priyojit. Today we turn our attention to a crucial aspect of eukaryotic DNA replication, the replication and maintenance of telomere. Linear chromosomes face a unique challenge, that is, their ends cannot be fully copied by the usual DNA replication machinery. To replicate viral genetic information and ensure chromosome stability, cells use specialized structures called telomeres and a remarkable enzyme known as telomerase. This elegant system prevents the gradual loss of DNA during cell division and plays a key role in cell lifespan, aging, and cancer. Let us briefly explore how telomeres are replicated and maintained to preserve the continuity of life at the chromosomal level. What is telomeres? Telomeres are specifically the special protective structures present at both ends of the linear eukaryotic chromosomes. They consist of repeated DNA sequences, that is tandem repeats. These are the tandem repeats of the telomere and specific telomere binding proteins. Each telomere has a short single stranded 3' overhang of about 12 to 16 nucleotides. Telomeric DNA is rich in guanine and thymine. The telomeric sequence is repeated hundreds of times depending on the species and cell type. Why are telomeres necessary? Telomeres protect important genes at chromosome ends from being lost during DNA replication. They act like buffer zones that prevent damage to coding DNA. Without telomeres, chromosomes would become shorter after every cell division. What is the end replication problem? End replication problem refers to the inability of DNA polymerase to completely replicate the 3' end of a linear DNA molecule, resulting in progressive shortening of chromosomes after each round of replication. So the question is, why does the end replication problem occur? DNA polymerase has two limitations. First one is it can synthesize DNA only in the 5' to 3' direction, and secondly it cannot start DNA synthesis on its own, so it requires an RNA primer. At the 3' end of the lagging strand there is no place to synthesize a primer, so DNA polymerase cannot fill the final gap. As a result, a small portion of DNA is not replicated. If unchecked, this would cause chromosomes to shorten with every round of cell division. So, role of telomerase. To solve this problem, cells basically use a special enzyme called telomerase, and it was discovered in 1984 by Carol Grader and Elizabeth Blackburn, and both of them received the 2009 Nobel Prize for their discovery. Structure of telomerase. Telomerase is a ribonucleoprotein enzyme containing TERC, that is telomerase RNA component, RNA sequence complementary to the telomeric DNA, and it helps telomerase binds to the 3' overhang. TERT, TERT, that is telomerase reverse transcriptase. It is a protein subunit. It uses RNA as a template to synthesize DNA, and it works like reverse transcriptase. Steps in telomere replication by telomerase. So first of all, the binding of the telomerase enzyme with the telomere DNA. Telomerase binds to the 3' overhang of telomeric DNA using its RNA component or TRC. Then polymerization. TERT adds a 6 nucleotide telomeric repeat to the 3' end, and DNA is synthesized using the RNA template. Translocation. Telomerase moves forward to the new end and it adds to another repeat. So this cycle repeats many times. Result is the 3' end of chromosome is lengthened. Completion of the complementary strand. After telomerase extends the 3' end, primase adds an RNA primer. So DNA polymerase synthesizes the complementary strand and DNA ligase joins the fragments. And when RNA primer is removed, it leaves a short 3' overhang. Then telomerase and aging, what is the relation between the telomerase and aging? In actively dividing somatic cells, telomerase shorten with age. Example, at birth, there is telomerase is 8000 base pair, and during old age it is approximately 1500 base pair. So telomerase activity decreases with age. When telomeres become too short, cells enter senescence and they lost the ability to divide. So telomere shortening occurs with age, and telomere shortening may be a marker of aging and a programmed process contributing to aging. Telomeres and cancer. Cancer cells divide continuously. Many cancer cells show increased telomerase activity, prevention of telomere shortening. This helps them avoid senescence. In our lab studies, telomerase inhibitors stop cancer cell division. Problem: such drugs may also affect normal cells and it could accelerate aging process, so much more research is required. So as we conclude, remember that the replication and maintenance of telomeres are vital for preserving the integrity of linear eukaryotic chromosomes. By solving the end replication problem, telomeres and the enzymes telomerase prevent the loss of essential genetic information during repeated cell division. This finely tuned mechanism not only safeguards chromosome stability but also links DNA replication to cell aging, senescence and cancer development. Understanding telomere biology help us appreciate how cells balance longevity and control. It's an insight that is crucial for your undergraduate, postgraduate and NEET preparations. So keep revising, stay curious and continue uncovering the deeper logic of molecular life. Thank you.